Hi, Michelle Glass here. Welcome back to another one of our chapter 10 lecture series. Our topic for this video is that muscle fiber, which remember is a fancy name for muscle cell. To me, we're really just using this term because of the size and the shape of these cells. Remember that your muscle fibers are extremely large. They're extending the full length of the muscle from origin to insertion. So if you think about those muscles attached to your femur, they're essentially going the full length of your thigh, or the rectus abdominis, which extends from your sternum to your pubic symphysis, the whole length of basically your abdominopelvic cavity. They are cylindrical in shape, so think about toilet paper tube. They are multinucleated, which just means lots of nuclei. This makes sense. You have a super long cell. It's going to need to make a lot of proteins. Those instructions are, of course, housed in the nucleus. And your muscle fibers are developing from the fusion of cells called myoblasts. So this is happening during fetal development. And then a few of these cells are remaining as those myosatellite cells that we mentioned in our last video. Those myosatellite cells, remember, are available for help with repair if you have damage to those cells. When we're looking at our muscle fibers, you should have recognized your skeletal muscle in lab because of those striations. So as we investigate further, we're going to look at the structure of those striations. These are an important feature, not just anatomically, but also in our physiological discussions. So sometimes people will refer to your skeletal muscle tissue as striated muscle because those are so apparent. We have some special terminology to look at. So when we are talking about the skeletal muscle fiber or cell, we'll see that that cell membrane has a special name. It's called the sarcolemma. That prefix sarco just means muscle. And remember, we refer to our cell membrane also as the plasmalemma. So that was to help make this connection here. This sarcolemma is special because it's an excitable membrane, which means it can have an action potential, or what we're going to call in the future, I went backwards on that. It can conduct an electrical signal, which we'll call in the future an action potential. And then the cytoplasm of the muscle cell is referred to as the sarcoplasm. So again, here we're using the special prefix sarco to reference our muscle. We have extensions of that sarcolemma down into the middle of the cell. Those extensions are called transverse tubules, or T-tubules for short. The significance of this is it's bringing that action potential, that electrical signal, into the middle of the cell, not just on the outside surface of the cell. We'll also see a special modified smooth endoplasmic reticulum. It's assigned a special name, sarcoplasmic reticulum, or SR. The key job of the SR is to sequester calcium. Sequester basically means to store and hide. We will see the SR wrapping around our protein fibers inside the cell, and we're going to call those bundles of protein fibers myofibrils. I'm going to show you a picture from your textbook in just a few minutes so that you'll be able to start getting that visual. When we're talking about the SR, we will have enlarged chambers in the SR called terminal cisternae. That's the plural form. Just terminal cisterna with an A ending is singular. This is where all that calcium is going to be stored. We need to pay attention to the fact that in order for the sarcoplasmic reticulum to actually sequester the calcium, it has to have the ability to pump it out of the sarcoplasm and then stick it into those terminal cisternae. And so embedded in the membrane of the SR, you have calcium pumps. We're also going to see embedded in the SR voltage-gated calcium channels, and these are going to control the release of calcium Voltage gated tells us that it's a change of charge. It's that electrical signal that's going to activate or open these channels. Once we get the electrical signal to the SR, those voltage-gated calcium channels open, and the calcium is then released into the sarcoplasm of the muscle fiber. Myofibrils, as we mentioned, are those bundles of contractile proteins. They are going to extend the full length of the cell. They're also going to be cylindrical in shape. They are composed of those contractile proteins, which are referred to as thin filaments, which is composed mostly of actin, and thick filaments, which is composed mainly of myosin. So actin and myosin are going to be our two important contractile proteins. The myofibrils are composed of units called sarcomeres. So essentially on the whole length of the myofibril, we have sarcomere, 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 sarcomere. So it's basically our functional unit where all of our contraction is going to be happening. And then we've already mentioned, so just kind of refreshing, these myofibrils are surrounded by both sarcoplasmic reticulum and T-tubules. Okay, so here's the picture from our textbook. So I've highlighted for you the sarcolemma, which remember is our cell membrane. And notice there's little holes, and that sarcolemma is extending down into the interior. It's shown in yellow, those T-tubules. So our electrical signal is not just on the surface of the cell, but also penetrating down into the middle of the cell. Those T-tubules are wrapping around the myofibrils. The myofibrils are composed of the thin and the thick filaments. This is what's giving the striation appearance to the cell. Those thick filaments are the dark lines that you're seeing in the striated pattern. Remember, your thin and your thick filaments are your contractile proteins, actin and myosin. And then that blue tissue there is showing us, I shouldn't say tissue, that blue structure there is showing us our sarcoplasmic reticulum. Remember, it's wrapping around the myofibril, is sequestering or storing our calcium. That calcium is specifically in those enlarged chambers called the terminal cisterna, which is the singular version, or cisternae with an AE ending for plural. Notice that you have this section with these three important structures together. We call this the triad. The triad is the terminal cisterna, T-tubule, terminal cisterna. Now, both of those terminal cisternae are parts of the sarcoplasmic reticulum, so you can use that as your memory device too. Okay, that's it for now.